Our next speaker is Dr. Jyoti Mishra, who is the founder of the Neural Engineering and Translational Labs, also known as NEAT Labs, at UC San Diego. She is trained in computational, cognitive, and translational neurosciences and have expertise in studies of attention, learning, and brain plasticity, and have contributed to technology innovations in video gaming, cognitive training, self-regulation training, and brain-computer interfaces. At the intersection of neuroscience and digital tech, NEAT Labs R&D is informing personalized mental health care, education, as well as climate change adaptation. NEAT Labs innovates neurotechnologies to scalable brain health mapping, monitoring, and precision therapeutics, and research across the lifespan and across the spectrum of mental health and diverse local and global communities. Over to you, ma'am.
Thank you. Thank you for the nice introduction. And let me share my PowerPoint. And if I do full screen, can you confirm that you see it?
Yeah, it looks good.
Okay, awesome, great. All right, so people, please feel free to ask any questions during the presentation. And I believe this is about 30 minutes, so prepared for enough time, so we have some questions as well. I'm delighted to be here at this meeting. And we met with Dr. Srini a couple of years ago because he mentored one of our really super talented postdoctoral fellows, now project scientist, Pragati Balasubramani, and I'll be talking about some of her work. And she's soon to be joining as an assistant professor at IIT Kanpur. And so we're very excited about the work that she's done and for the future of her work. So at NEAT Labs, we are studying cognitive brains as a portal between the experiences that we have. Every new experience in life shapes the way our brains change at that point in life. And that in turn shapes our executive and mental health behaviors. And so we are constantly looking at this relationship between brain's experience and mental health. So let me tell you what we're doing in the lab. When we started off our group is a young group in that we only started our labs about 4 years ago. This is our 5th year. And one thing we really wanted to do was to enhance neurotech for mental wellness. And what that means is that in order to understand mental health better, we said, well, we want to make brain mapping and tools that are related to brain mapping more accessible to our community so that we can do this more easily across questions. Go ahead. Does somebody have a question? Okay.
No.
Okay. All right. And how those brain targets that we define can then identify aspects that would then lead to techniques that can inform mental wellness. And when we understand mental wellness, we want to understand that in the context of larger data. So we want to be understanding circuits. We want to be understanding how the individual is behaving, how if the study requires us to involve the family, what the family context is like, what the community context is like, and then more details at the neuronal level, what changes are going on. That is something that our co-director, Dakshin Ramanathan, does very well, and he's going to talk more about that in his talk that's upcoming. So we're in the Department of Psychiatry, and what really drives our work is really how neuroscience can inform mental health better. And this is a review from Nature in 2012, so it's been a while, but the concept is that for any kind of neuropsychiatric dysfunction, there is cognitive impairment that's underlying it. And so to understand cognition better and the neural processes that support cognition is very important. But if we look at how mental health care is practiced today, it doesn't really inform, it's not really informed by how neurocognitive dysfunction happens. If you look at this little cartoon here, usually people just look at the behavioral symptoms that are exhibited by the individual, but not beyond that, what is the neurology behind it? What is the neurocognitive status behind it? That's not really done in psychiatry too much. And so what we said was, to achieve this integration, our method needs to be more scalable, in that if we kept doing our methods in our lab, this is not going to cut it. It has to scale to the clinical and community-based settings. It has to be cost effective because for the same reason, it has to be scalable, and it has to be rapid. Our doctors don't really have time to get into methods that are going to take a day long. They have probably 15 to 20 minutes with a patient. And it has to still be comprehensive. We can't really have the luxury to say, okay, let's just measure one cognitive domain, such as attention, but overlook other domains that may be important for the individual. And then, of course, the idea is to be data-driven instead of just subjective symptom-driven. Okay, I think someone may be off mute. And if you guys could do a mic check, it might help the rest of the audience as well. All right, so this, about 2 years ago, we developed this platform, the Brainy, or the brain engagement platform, which for the purposes of doing comprehensive brain mapping. And it's a rapid assessment suite, and it consists, it's built off of a game engine, so so people do it in an engaging manner. It's like takes 30 minutes to do. The point is that there are many exercises in there that revolve around different aspects of cognition related to conflict processing, working memory, internal attention, response inhibition, emotion processing, reward processing, and so on. And this is how, on the left is a picture of what it looks like when an individual is doing this assessment. And the assessments in there are actually standard cognitive assessments that the literature has published on, are important for understanding cognitive status across different populations and across the lifespan. So the idea was, can we have a standard set that can be done fast for people that are from, kids who are 4-year-olds to older adults who are 90 years old or more, and use the same standard set. And actually, Dr. Srini's lab is very aware of the details of these, each of these, and these are actually, it's published data now. So if people are interested in looking at what is each assessment, they can look at the published work. So here, our wonderful team was then able to extract these kinds of cognitive performance reports for every task. So for any given individual, one can get what their cognitive behavioral performance is. And then this is very traditional kind of processing that we understand in EEG space that underlies cognitive domains. So if you're familiar with EEG, these plots are showing spectrotemporal data, and FCZ. So FCZ is an electrode that's in the frontocentral location of the head, up here about, and on the scalp view, you can also see FCZ will be somewhere up front in the middle over here, and parietal occipital is on the back over here. This is on the right are the spatial topographies, and on the left are basically how the spectra are represented temporally over time, and also spectrally from the relevant frequency bands. So the relevant frequency bands are theta band that goes up to 8 hertz, then alpha band that can go up to 12 hertz, and then beta band. And what we see is that yellow represents excitation, so event-related synchronization that's happening when a stimulus is presented, and that's very standard for such tasks. Basically, in the brain, whenever stimulus is presented, there is a bunch of neural firing that happens, and that leads to synchrony in theta alpha oscillations, and that's what's being represented here. These are task-evoked oscillations, and beta is a desynchronization that is also known to happen post a stimulus presentation. And that's what's being represented here, which is data at the scalp level. One thing that one of our graduate students, Ale, who's actually in this picture, did was also enhance the processing of data in Cortex. So this is a pipeline that he developed that we've now standardized for analysis in the lab. That's sort of an innovation that other labs are starting to use, but we're the first to use it, in that he enhanced the way scalp data is represented inside the brain. So if you look on the right panel here, there are existing protocols for of what's called source localization, inverse mapping methods. If you're not familiar with inverse mapping, think about how, say, when an earthquake happens on the surface on the surface, when you feel it, you feel a perturbation, but the source of that earthquake, the epicenter, is somewhere under the earth, and there are techniques such as beamforming that can be used to actually say, given the resistances of the different layers of the earth, where would the underlying activity be coming from? And it's the same exact style of process that happens for brain data as well. And in this case, what Ale did was really this bottom plot, the hyperparameter plot on the right side here, shows that what he was able to do was apply some biological constraints to the source mapping. That, you know, sources can't be outside the brain. They can't be because of you know, muscle noise or from eye movements and so on. So better representing physiology, especially with regards to noisy physiology. He was able to refine the mapping, and what we get here is that you see how there's a focal point here that's in red, better than on the left side here. The cortex doesn't really have a focal point in this case. So he's able to produce these methods that are better for cortical localization. And this is an example of how we've used that kind of method across tasks. So for example, this is the stimulus evoked activity that's happening in the theta band across different tasks that are run on the Brainy platform. And you can look at how there's yellow means synchronization of activity that's happening. And across tasks, you can see this activity, and then you can further statistically correct for this activity. Again, something that people haven't done in the past. And on the right are the statistically corrected plots, which are still showing very strong activations across different sites in cortex. So, so this kind of methodology is exciting for us because it takes a very scalable method, such as a low-cost EEG that can be applied rapidly in clinical settings. And now we have these brain maps that can be compared to what we know from the neuroimaging literature, such as with fMRI. Of course, our spatial localization is an approximation. It's not as accurate as doing an fMRI, but then an fMRI equipment costs $1 million. So given the trade-offs, we can now implement this not just within our local communities, but also we have work happening in locations in India and in some parts of Australia, which is basically because this is because of the scalability of the methods. So this is Pragati Balasubramani's paper. As I mentioned in the beginning, she's an alumnus of Dr. Srini's lab. And we're very proud of the work that she did pioneering this application of this source localization method. And she showed that across sessions, so if you were to do Brainy recordings in healthy people a week apart, you have high reliability. So 2-visit reliability is shown here. So all the regions that are colored are showing high reliability. There are some frontal regions that don't have high reliability, but sometimes that's natural because frontal regions can have cognitive fluctuations from day to day. On the right-hand side, what we were measuring here was plasticity is Brainy sensitive to anything that you do to the brain. So in this case, we were applying neurostimulation, so transcranial magnetic stimulation, and the protocol is shown on this side so that you can have brain stimulation that's of type A. I don't need to get into the details of what type A and type B are. If anybody has questions, they can look in the paper. But the point is that we knew that one kind of stimulation was slightly more effective than the other, A versus B. And then we are able to contrast and see that kind of sensitivity in different brain regions and in different frequency bands on the Brainy platform. So it's not only, the point was that it was not only reliable, but also sensitive to change when we apply a change on the brain surface. Okay. And then where we're going with this is that this was, the year just prior to the pandemic. We've continued our work during the pandemic as well. But obviously, work has slowed down a bit. But even just very easily in the year prior to the pandemic, we had 300 people come into the lab to do these do these recordings. Very easy to do. So participants would be very easy to have participants come through the research. The research logistics were easier. And what we then can start to formulate is standardized neural processing that underlies cognition, not just, for a single study. Usually neuroscience studies will have very small number of participants, but now you can have larger and larger number of participants and create norms to say where there might be deviations in a given person. So the colors here are representing mean activity is in green, while other colors are being represented in if you have activity that's greater. This is for one person. So if you have activity greater than the mean in any brain area, then it's represented in red. And if you have much lower activity, so hypoactivation, which can be concerning for dysfunctional cases, and that's shown in blue. So let me just, okay, I'm good on time. So Dr. Srini and others, if I should stop before 6:30, I'm happy to. 6:30 is in our time. I think we're 17 minutes, 18 minutes in, and I'm happy to take questions earlier. So please let me know when to stop. And then again in this paper, what we were able to do was to relate aspects of brain function. So this is in dorsolateral prefrontal cortex. It's very well known to be a site of dysfunction in various psychiatric disorders, and we were able to confirm this in the study. Although even this work was conducted in healthy participants, even a healthy person can feel less or more mentally well on a given day. So it's a continuum. It's less of a either or status, patient versus not. This is more of a continuum. How do I feel today? So that kind of a mental health status, when we measure that in healthy people, if you have lesser activation in dorsolateral prefrontal cortex, you see lesser, you see poorer mental health as well. And this was seen in the PFC and then also seen in parahippocampal cortex. So the point here was really that we could relate aspects of starting to relate aspects of mental health to underlying cognitive brain function. Another study that the team led this year was that now we can ask on a larger scale aspects that are interesting for mental health behavior. So for example, in this study, we were interested in looking at aspects of social isolation, loneliness, how a person feels isolated, because loneliness can be a big trigger for developing clinical mental health symptoms. And then on the contrasting side, there are aspects of wisdom that some UCSD labs here have and who are co-authors on the work have shown to be very important for having resilience through the lifespan. So the measures that go into resilience are aspects of pro-social behavior and empathy towards others. And wisdom is actually divergent from intellectual ability, crystallized intelligence. So it's not about how intelligent one is, but it's more about, you know, the life experiences that have shaped the way you think and the way you interact with people. And so it turns out that this is work that we confirmed from prior behavioral work, that loneliness and wisdom are inversely related. When you feel more alone, those people tend to have lower scores on the wisdom scale. But on the neurocognitive side of things, nobody had really worked at, well, is there a brain and cognitive basis for these phenomena? And what we were able to show here was that in one of the tasks, so for example, this is an emotion bias task. And what happens in this task is that you're supposed to do a very simple task where you have to say which way an arrow points, either left or right. And behind the arrow, you will see that there is a face that's presented. That could be a happy face or a neutral face or a sad face or an angry face. So what we found is that the lonelier individuals actually were more sensitive to angry faces than neutral faces. They have more angry bias, and their processing of a simple cognitive task would slow down if an angry face was shown. And this was supporting a theory in the literature that there's a social threat theory, that people, lonelier individuals don't really engage with others because they may find others to be more threatening and have a bias towards, not engaging with others because, like what I said, that they appear threatening. And so we confirmed this bias, whereas on the wisdom side, individuals who were wiser had a bias towards happy faces. And in this case, their processing was quickened and more alertness was observed when there were happy faces presented. So this was interesting. And we were also able to go in and look at brain structures and brain function, and showed that areas that are in, such as the temporoparietal junction, that's involved in theory of mind, and areas of attention are involved in biasing for angry faces, whereas on the other side, for the happy bias, we saw again theory of mind and insula. Insula is involved in developing more affective regulation and insight. So the brain areas were very much aligned with the theory of how loneliness or wisdom might be represented in the brain. Okay. This was yet another study where we're looking at, remember that we could look at work across populations when you're doing recordings across the lifetime. So in this case, we can look at differences across populations. In this case, the study that we're doing is younger versus older adults. How does processing get represented? So younger adults show greater processing in frontal areas. So the more red or orange processing that you see is being represented in younger adults. Whereas on the other hand, in older adults, there's more processing in areas that are related to what's called the default mode area of the brain, which is more related to task unrelated thoughts or mind wandering. So it was interesting that older adults had more performance was being depicted by greater activation in brain areas that were related to task unrelated processing, and that may be the basis of their poorer performance. So older adults tend to do worse than younger adults. Okay, and then one of the last slides I want to show you is now what we're doing is that, okay, we've developed a platform, and where we want to take this is to embed it within interventional work. And so this is a study where we are using some of our digital interventions, which I might have time to get into at another point today. And sorry, not today, at another point in this meeting, maybe another year, what do our interventions tell us? But essentially saying that when you have, when you provide these kinds of standardized trainings, you're changing the brain. And how could Brainy be used to really measure these changes in brain function? So these studies are looking at aspects of depression, burnout, and also working with healthcare physicians who we know have been very much affected, especially during COVID. We're also conducting a study that is looking at these changes in dorsolateral prefrontal cortex due to brain stimulation. And so what are the changes that happen pre and post? And again, this is in depressed individuals. And finally, some of our prior work has shown how aspects of internal awareness and internal regulation are very powerful in terms of improving mental health, much more so than aspects of being able to do external tasks and so on.
Hello, please mute yourself. Hello. Participant should mute yourself.
Okay, so so in terms of internal regulation, so we developed a digital meditation type protocol that not just a single individual, but also parents and children can do together. So all of this work is actually ongoing. And some of this work is, Pragati is still involved in some of this work, so I'm sure she'll be able to tell you about it at some point in time. And with that, I'll just come to the research summary of the Brainy platform that has allowed us to scale cognitive brain mapping to labs, clinics, and community settings to better understand sort of the brain bases of mental health behaviors. And then current studies are engaging Brainy within intervention protocols. We develop our own digital trainings and standardize those as well. And then the point is that we want to be able to enhance well-being. And one of our more sensitive targets has been depression, which has a very high worldwide burden. So we're very excited to work in that field and maybe move the needle in depression care. So that's all we have for you today. So in essence, Neat Labs develops technologies for mental health, and our teams are very widespread. We have a team here that's also been online, and we also have had an active team in the past in India, and also some work that we're continuing in Australia. So thank you for your time. I'm happy to take questions. And also, induction will be starting shortly if, people don't have questions.
Any questions?